Okay, so today we're going to talk about the liver. And in the body, this is where the liver is located, right underneath the heart. And this is what the liver looks like on the outside. It has 4 lobes. And this is the internal structure of the liver. So basically, the liver is supplied with blood from the hepatic portal vein and the hepatic artery. And the hepatic portal vein brings absorbed nutrients from the intestines, and the hepatic artery brings oxygenated blood to the liver. The hepatic vein brings blood to the vena cava and to the right atrium of the heart, but it brings deoxygenated blood. So within the liver, the hepatic portal vein brings some toxins, high nutrient levels, and low oxygen levels because it's already been through a capillary bed. And the hepatic vein brings away nutrient levels but less toxins. And and so that's the structure of the vein supplying it.
And so basically, for the liver structure, the liver consists of 4 lobes, and these lobes contain many microscopic lobules. And these lobules are specifically structurally adapted to their functions. So we're briefly going to talk about the structure of the liver and the lobules. Audrey will talk about the structure of the hepatocytes, and then we'll go into its functions. So the lobules in the liver are each surrounded by branches of the hepatic artery and the hepatic portal vein. And down their center runs a branch of the hepatic vein. In the lobules, there are these specially adapted capillaries called sinusoids. And basically, these capillaries connect the hepatic portal vein and the hepatic artery with the hepatic vein. And they are special capillaries because, firstly, their lumen is larger than that of a regular capillary. Their walls are very porous for easier diffusion. They consist of one layer of cells with many gaps. They have no basement membrane, and they receive blood from both the hepatic artery and the hepatic portal vein. This structure allows all components of blood to be in close contact with the hepatocytes for processing. And a lobule also has bile canals running from the center of the lobules to the bile duct on the outside to secrete the bile. And also in sinusoids are Kupffer cells, which we'll look at more in depth in the functions, but they basically removed all erythrocytes, and macrophages are also present to remove pathogens.
So hepatocytes are the names of the liver cells, and basically they have microvilli on the lumen side, to increase surface area for diffusion. They have a plasma membrane that has a lot of transporter proteins and insulin receptors to regulate the glucose level in the blood. And they have a large rough ER for protein synthesis, a large Golgi apparatus for preparation of secretin, and they have a lot of glycogen granules and fat globules for energy storage. They also have a lot of mitochondria for energy production, and large peroxisomes, or vesicles containing catalase and other digestive enzymes for detoxification of poisons and alcohol. So now that you know the structure, we're going to talk about the functions of the liver. Basically, the functions include...
Okay, so the liver has actually over 500 functions, which can be summarized in 10. So firstly, regulation and storage of carbs, fats, and proteins is one of the main ones. Storage of vitamins and minerals, excretion of nitrogenous waste, synthesis of cholesterol, synthesis of plasma proteins, and detoxification, as well as inactivation of hormones, thermoregulation, breakdown of hemoglobin, and bioproduction. So these 10, I guess, are obviously like major ones, which have been summarized. However, they can be further broken down into, I got 7 functions, and we'll be starting with regulating nutrient levels. So the way the liver regulates nutrient levels is, firstly, as we've mentioned before, the hepatic portal vein transport digested nutrients from villi of the small intestine to the liver. The concentration of nutrients in this blood will vary depending on the meal. If there's a high concentration of nutrients in the blood, this can lead to damage of other organs like the brain. And so the hepatocytes will detect the high concentration of nutrients and will absorb, store, or release nutrients as necessary to maintain a constant level, always maintaining a balance.
So the storage of nutrients, is that what you just did?
I did regulating nutrients.
Okay, so lipids and carbs can be stored in the liver, and it's called glycogenesis.
Glycogenesis. Wait,
Glycogenesis.
Glycogenesis.
Okay, so upon stimulation from insulin and that was released from the pancreas, the hepatocytes absorb glucose from the blood and store it as glycogen. And glycogenolysis. So that's when the stimulus is from glucagon from the pancreas, and glycogen is converted back into glucose and released into the blood. So this regulates the glucose levels in the blood, and this way glucose levels are kept between narrow limits and homeostasis can occur. So vitamin A and D are also stored in the liver, and iron is stored as ferritin in the liver, and copper and cobalt are stored as vitamin B12.
So on top of all this, the liver also plays a major role in synthesizing nutrients in the body. So, for example, the liver can synthesize non-essential amino acids by transamination. It produces plasma proteins like prothrombin and fibrinogen, which are required for blood clotting. It produces albumin, which regulates blood osmotic pressure, produces globulin, which helps with the transport of other molecules like insulin and lipids, produces lipoproteins to be used to transport cholesterol and lipids around the body, and finally synthesizes cholesterol. So cholesterol is required in cell membranes to make vitamin D and hormones, and the liver produces cholesterol from glycerol. Around the body, cholesterol is transported to body cells in the form of LDL, and the liver can also remove excess cholesterol. And the way this happens is that when there's excess cholesterol, it is not transported as LDL around the body. It is transported in HDL. And the hepatocytes remove this HDL from the blood and convert it to bile salts.
And basically a diet with high fat content leads to high concentrations of LDL in the blood, which is the bad cholesterol. And also concentrations of LDL or a low concentration of HDL leads to heart disease. Okay, so the liver also detoxifies toxins that enter the body. So enzymes present in the previously mentioned peroxisomes of the hepatocytes detoxify poisonous substances such as hydrogen peroxide, drugs, and alcohol. The toxin is chemically modified to make it less harmful. Chemicals added to make it more water-soluble, and they also modify the toxin and is released back into the blood to be filtered by the kidneys and excreted in urine. Some of the toxins that the liver detoxifies include alcohol, chloroform, and DDT. Yeah.
Okay, so we've also now got breakdown of hemoglobin. So erythrocytes, on average, have a lifespan of 120 days, and upon their death, they either rupture, releasing hemoglobin, or are removed by spleen, which also releases hemoglobin in the blood. The Kupffer cells, previously mentioned in the sinusoids, absorb the old worn erythrocytes or the hemoglobin by phagocytosis. The copper cells split the heme group from the globin, and the globin is hydrolyzed into amino acids to then be released into the blood. The iron is then removed from the heme group, forming a yellow substance called bile pigments or bilirubin. Bilirubin is released in blood, where hepatocytes pick it up and secrete it into the bile. In the meantime, the Kupffer cells release the iron into the blood. This is where the iron combines with plasma protein called transferrin to form a transferrin-iron complex. The bone marrow cells will absorb the transferrin-iron complex and make new erythrocytes out of it. Finally, the hepatocytes take up the transferrin-iron complex and store it as ferritin.
So another thing that the liver does is produce bile. And also the bile is produced in the liver but stored in the gallbladder. Bile consists of 90% water, bile salts made by the liver from cholesterol, amino acids, and salt, ions like Na+ and Ci-, cholesterol and phospholipids, sodium hydrogen carbonate, which neutralizes stomach acid, and bilirubin.
Okay, finally, the seventh function is excretion of nitrogenous waste. So in the body, excess amino acids and nucleic acids cannot be stored. This is how amino acids are deaminated, whereby the NH2 group is removed, and the amino group is converted to ammonia, combined with CO2 to form urea. Urea is then released into the bloodstream and removed by kidneys. The rest of the amino acids are used in cellular respiration.
And so basically, alcohol damage is something that happens to your liver when excess alcohol is absorbed. And alcohol is basically absorbed by the liver and converted into other non-toxic substances, but excess amounts causes damage. The first stage of alcohol damage is fatty deposit built up in the liver, which can cause alcoholic hepatitis. Alcoholic hepatitis causes inflammation of the liver, leading to nausea and jaundice. The second stage is the death of liver cells and the formation of scar tissue, or cirrhosis. Liver cells loses its ability to function and can lead to liver failure and then death.
Jaundice is another disease which may be caused by liver malfunctioning. So essentially, it's caused by excess bilirubin in blood. Very high levels of bilirubin in blood can also lead to brain damage. The symptoms of jaundice are yellow skin and yellow white of the eyes. In this respect, infant jaundice is often found in newborns because higher level of bilirubin may be found in their bloodstream as baby blood cells are replaced. The liver is not yet capable of breaking down this bilirubin, so infants often have jaundice. Treatment to jaundice involves blue-green light, which breaks down bilirubin, and adult jaundice is caused by problems with the liver.
So yeah, that is all the functions, the structures, and diseases that come along with the liver. Thanks for watching.